All right, I see it's 4 o'clock, so I think we're going to go ahead and get started. I'll kick things off by doing some introductions, so anybody else joining on has a minute for catching up. So good afternoon, everybody, and welcome to our Summer Disease Management webinar. I'm Mike Bastow, and I'm a tree fruit specialist with the Eastern New York Commercial Horticulture Program, and will be your host today. I'm also joined by my colleague Dan Donahue, who's down in the Hudson Valley, and we're also joined by our Champlain Valley technician, Andy Gallimberti. Before we begin, I'd just like to briefly orient everyone who might be new to using the Zoom platform. So if you haven't done so already, please double check that you're muted and that your video is turned off, and you should be able to do that by hovering over your screen, finding the toolbar, and clicking on these icons. You should also be able to find the chat box, which you can access by this chat icon. By hitting that toolbar, that should pop up the chat box. And in that box, you can enter in any questions that you have as we go along. And then at the end of the presentation, Srdan will take a look there and be able to answer all of your questions. So with that, our speaker this afternoon is Dr. Srdan Acimovic. Srdan is a senior extension associate in plant pathology and is located at the Hudson Valley Research Lab in Highland, New York. His research and extension program is focused on improving our knowledge of plant pathogen biology, ecology, and population dynamics to facilitate the development of new disease management strategies. His goals include elucidating quantitative parameters of pathogen survival and stress, improving our understanding of plant defense responses under field conditions, and developing new or improving on existing strategies for disease management. Two integral parts of his program include fungicide and bactericide efficacy trials, and extensive outreach to deliver disease management information to directly benefit the apple industry of New York. So with that, I'm going to turn my screen over to Srdan.
Okay, good afternoon, everybody. I'm just going to go and hit it right off. Thank you, Mike, for organizing this webinar on such an interesting topic that I think is going to be very relevant this year. So I'm going to start first with just briefly showing you here the symptoms of some of the pathogens that we're going to cover and talk about today, and some of the challenges in control, and also reflecting on the new findings from my lab, and also some of the data from managing of these diseases. So this is an important topic because every second or third year we have big issues with certain, of these diseases, and this webinar seems to be well timed at this time of the year. So I'm going to be covering these 4 or 5 pathogens that you see in blue, including black white rot and bitter rot and Sooty and fly speck and Marssonina leaf blotch, with purposely leaving out gray mold because this disease is intermittently showing the botrytis cinerea, and in the interest of time, I'm not going to be covering it today. And in general, these pathogens are interesting because they can be present both in the field and in the storage and become storage decays. So they are double players in that regard, and that's why they're so difficult to manage in one regard. So I'll first open and talk about the first group of pathogens, which is going to be, in this specific case, black rot. This is a list of symptoms of black rot that you can see here. I would like to just briefly emphasize to you that the pathogen in this specific case is very well known to overwinter on these big cankers that it causes, that you can see on the left side. And then also it overwinters very well in the mummified fruit, especially the ones that are left after thinning agents that sometimes cause higher percentage of mummified fruit that don't fall off. And typical symptom of the frog eye leaf spot that you see here on Cortland, and sometimes these brown or black patches that look like specks sometimes where the infections happened. But the most common one that you would probably see besides the frog eye leaf spot would be the symptom on the fruit. And this rot specifically is quite a firmer rot. It's spherical, so meaning that the fruit does not change shape. and it's a drier rot. So when you cut into it, you don't see too much soaking of the tissue. The frog eye leaf spot can become severe, very close to the cankers, which you see on the left, and sometimes the leaves can cup and curl. Giving you an idea that the canker might not be actually from fireblight, but actually from black rot. So this is the life cycle of the pathogen. On the left top corner, you can actually see these pear-like structures with an opening on the top, which is basically the sexual reproductive structure that we call perithecia, in which we have ascospores that are spores, formed by sexual reproduction, which are ejected in the spring, along with the other fruiting body that is right next to it, which are pycnidia. And conidia, asexual spores form in them, and they are also released with rain, and they can be splashed or wind-driven to new leaf tissue, such as leaves very close to the fruit mummies or cankers that actually hold these fruiting bodies. Once the spores reach the leaves and fruit, which you see in the top middle part of the graph, you can have infections leading towards the symptoms of frog eye leaf spot. Sometimes, rarely, you can see infections on wood as well. And eventually, the symptoms occur on fruit, and the rots usually lead towards desiccation of the fruit, and basically you see the fruit as a fruit mummy. And that fruit also, along with the cankers on the wood, serves as a source of overwintering inoculum. So basically the brush on the ground and also the dead cankers on the big limbs on the wood. Anywhere where there was damaged wood, this fungus inhabits and can actually overwinter there as mycelium, which is a hair-like structure in which the pathogen grows, and then also as perithecia and pycnidia, these two fruiting bodies that you see on the left middle side of this image. So this is the way how pathogen basically cycles every year. It can have multiple cycles of release of these conidia, and only one cycle of ascospores being released. And besides the overwintering that I mentioned to you, usually what happens is that it can also be present on the trunk and dead brush. So good sanitation in the orchard is essential to get this pathogen under control and make sure that your fungicides are highly effective when you reduce the inoculum. The infection usually happens if the infection pressure from last year is high. It can happen as early as early spring. But in most cases, it's usually when the warm rains start during the spring. And as I said, the sources of inoculum are perithecia and pycnidia. Most of the time, the period that usually people start looking at them because the fungicides become a little bit more sparse at the time is from petal fall to harvest. But in the untreated checks that we have at the lab here in Highland, we can see infections as early spring because we don't treat those trees with any fungicides. So it can also cause dormant infections, meaning that after the infection has set in, the pathogen lays dormant for some time, and then it reactivates with the fruit maturation. And it seems that fruit ripening definitely allows pathogen to be reactivated and cause the rot. And it's definitely a pathogen that is a minor issue in many orchards. It is endemic. It's present in almost all the orchards, but is largely controlled by different fungicides that we apply for scab and for powdery mildew and other issues that we have early in the spring. So thinning can contribute to formation of the mummies, which this pathogen inhabits. And eventually, sometimes you can have lenticel issues with infection from this pathogen on semi-mature fruit during the summer that some people actually can mix up with other causes of damage, such as phytotoxicity and lenticel breakdown issues. And these lenticel spots sometimes are deceiving, but they are actually, in some cases, black rot. And then later, if these are not taken care of, the fungicides are not, fungicide coverage is not maintained, these can continue to develop and decay in storage. And lenticel spots, can be caused not only by black rot, by the white rot fungus, which I'm going to talk next. And also in very rare cases, bitter rot fungi from Coleoptracum genus can cause those small spots. So it's kind of difficult always to diagnose those spots and, and to pinpoint them as a direct cause. So I'll briefly switch now to white rot. I wish to emphasize to you that white rot is a disease that carries the name, of course, of the symptom on the fruit, which you see on the right side. And this white rot is quite more wet and soft and soggy and drippy in comparison to the black rot. But sometimes the symptoms can be similar. It can be very similar in dry parts of the year, so you can't really distinguish them. But one thing that really stands out is the symptoms you see on the left side, the purplish to brown patches you can see on the bark. This is actually caused by Srdan Acimovic, which is an opportunistic pathogen, meaning that it requires the tree to be weakened by some abiotic or biotic factor. And then in those cases, the tissue that is weakened is a perfect tissue for this pathogen to inhabit. So it lives and dwells on the surface of the bark as an opportunistic fungus. And then if it does see an opportunity by weakening the immune system of the plant, it moves in and causes these patches. And eventually you can see these cankers that sometimes might deceive you that this might be from fire blight, but actually this is just because the trees were not taken care of very well or irrigated to allow good health of the tree, and then allowed these cankers to form. The white rot fungus is caused by the fungus. The white rot fungus is called Srdan Acimovic. It also overwinters in the same mode as black rot, as perithecia and mycelium and conidia in the pycnidia as fruiting bodies. It causes infections during the hot summer days. And as I mentioned, the rot is slightly different from the black rot, being more drippy and more soft. It usually causes irregular margins in the fruit. And what is really interesting is that sometimes when it's drier, white rot can be very similar to black rot. But usually, if you cut the fruit, you can find that the rot is more of a cylindrical shape, which would indicate on a white rot versus the black rot. So it causes also these bark cankers that I mentioned to you, and that's also where the pathogen survives and overwinters. And that flaky bark on the trunk is sometimes associated also with herbicides. And the way how probably this works together, based on research that has been done in the 80s, is that most likely the herbicides that hit the trunks because of the nozzles that are not directed to the ground might allow desiccation of the bark, which then stresses that bark tissue and allows this pathogen to become more dominant on the bark and causing these patches. And it seems that whenever the trees are stressed because of the lack of water or desiccation by the herbicides, this pathogen gets really active and reactive, and then causes these black patches. So, and it seems to be that research is lacking there yet to prove that there is a connection between herbicides and Srdan Acimovic, but there's a definite connection between the drought stress, meaning if the trees are not in their not really well irrigated and irrigated on time, would allow this pathogen to become something that might cause more issues on the bark and on the trunk. How would you approach the management? Well, first of all, as I mentioned, the good orchard sanitation is important. You should definitely remove the brush and burn the brush or bury it if you can. Definitely cut out the cankers and remove the mummies if you can from the trees. And especially do that during the time of winter pruning when there is a chance to do that. Just leaving the brush or the mummies on the ground is not going to help because the inoculum is right there. So it needs to be taken out or buried. If you can, you should avoid putting stubs because any damaged tissue or stressed tissue with stubs there might be a perfect environment for these fungi to inhabit that tissue and allow inoculum to be high up in the canopy, allowing infections. If you can, when you're picking, you should prevent fruit bruising because the pathogen can be spread from the other fruit that you're picking on the new fruit. We are largely dependent on fungicides to control these fungi, and the FRAC 11 group, or the group 11 that you would see on the label as a number, the quinone outside inhibitors, which would be your Flint, Pristine, Maravon, are the ones that are very effective against these fungi, both of them. And thiophanates, which is your topsin or thiophanate methyl, is very effective, and also the Captan is very effective against, because it's a multi-site contact activity, and you should always mix those two against these fungi. But if I would go and list the top choice products, definitely Topsin M or Captan would be a really good choice. However, remember that even though it does control black and white rot, it doesn't. Topsin is not really known to have any control, or really satisfactory control, even close to being satisfactory on bitter rot, which I'm going to talk about next. But if you see this list here, anything from Flint, Luna, Sensation, Maribon, Sovran, and Pristine are very effective. And then you should always apply these products along with Captan. Primarily, these products are applied in, usually to control bitter rot. And then somehow, they are sometimes used against powdery mildew, because the QOI fungicides that I mentioned, the FRAC 11, are very effective against powdery mildew. So if you had issues with powdery mildew last year, some people would use these products at pink or petal fall, and in that way contribute towards the control of black and white rot. But being that that the bitter rot is more bigger issue, these fungicides are effective against bitter rot, and we're going to talk about that later, and also very effective against Sooty, Blotch, and fly speck. We tend to reserve them for the cover sprays later in the season to control bitter rot primarily. So one thing that we do recommend, at least in the Hudson Valley region, is that we usually never recommend that a grower should surpass the deadline around the 10th July, because if the fungicides that are effective against the rots are missed from that date on, it seems there is more issues with different rots. And that's just based on historical weather conditions. If you do apply these fungicides, really you have to recover every time when you get accumulated 2 inches of rain in a single event or multiple events. And the residue has to be maintained on fruit until harvest. So some of the late varieties, such as Pink Ladies and Granny Smiths, if anybody grows those, or Golden Delicious, they need to be covered up until the time when they're harvested. Especially organic orchards they're definitely the place where these are an issue. And people are really dependent there on liquid lime sulfur for Sooty, Blotch, and fly speck control. In those cases, the addition of oil to liquid lime sulfur allows the damage to fruit skin, which then in turn leads toward more, of these fungi, black rot and white rot, like to establish more there. So in those cases, it's a tough call when you have an organic production. In general, if we do have injury on the fruit surface, that's going to contribute to the worsening of rots in the orchard, and you should avoid trying to have that. So any insects that can be present there are not welcome, because they contribute also to the spread of these diseases there too. So now I'm going to switch and talk about the bitter rot, which seems to be an emerging and rising problem, especially south of Albany, and in some pockets where in Western New York, or where the Lake Ontario is. And this is an issue which is quite different from black and white rot, because it's caused by a different pathogen called Atotricum genus. And in there you have multiple species that cause this disease. And so far we have been operating in the Hudson Valley under the opinion that most likely the species that is dominant causing bitter rot is called Atotricum fiorini, although we did not have any evidence so far. We now have that evidence, and most likely that's the dominant species, but we did not really, we just don't know whether there's more species, because we do see a difference in symptoms. There's slight variations, as you can see here, not only from the early infections, but also there's difference probably because there's multiple pathogens causing this disease. But the typical telltale sign is if you cut the fruit, and then you can see this typical V-shaped wedge necrosis that you can see on the right lower image that I've presented. And also when you look at the this rot from the side, it's flat as a flat saucer, which is basically indicating you that it's most likely bitter rot and not a black or white rot in case. And sometimes, of course, when there is wet weather, when there's rain, these concentric rings that you see in the middle of the rot spot where it started, you can see these orange small droplets that actually are huge amounts of spores of the pathogen, asexual spores that allow the pathogen to be rain splashed and dispersed to new fruit that can be infected. So it's a growing issue in New York because we had outbreaks in 2015 and 16, and mostly it's around in the Hudson Valley region, but also people in Canada, in the Ontario region have issues with it, but also in Pennsylvania and other southern states like Kentucky and North Carolina and South Carolina and Virginia. So this is not an isolated issue that is just happening in New York. It's definitely widespread on the Eastern Board. It seems that that many growers leave out their fungicide spray sometime during October with the late varieties, and closer to harvest there's no coverage by fungicides, and that's the way how this fungus moves in and then is being carried into the storage. It seems that the stem end of the fruit is the place where water keeps after the rain, and that's where this pathogen can infect, but the symptoms don't show up until you move the fruit into the pre-storage area. And then, while the bins are waiting to be placed in a cooler, they can start progressing. And then you put the bins inside, and then once you start packing, you see losses up to 5 to 7% in storage that we have seen in 2016 from bitter rot. So, and we have also seen in the field losses up to 60% on Honeycrisp in some orchards in the Mid-Hudson Valley region, and organic production in the southern part of the state that went up to 100%. But if we would average all these, I think they would be around 20%. 20 to 25% would be the average that we have seen across the state. Certain years we did not see outbreaks, but the fungi are present. We have seen some orchards that have not been sprayed, so it's there. It's just, maybe the people are spraying really well. And we have seen outbreaks in 2018. So why do we have more issues with bitter rot now? If you ask Dave Rosenberger, who has been in my position in the last 30 some years, the most important trick that led to more issues with bitter rot is the implementation of 77 days of pre-harvest interval for mancozeb, because mancozeb is very effective against bitter rot. We do tend to have in the last, I would say 5 to 7 years, more hot and humid summers with very warm wetting events that come with it, which is the weather that really favors these pathogens to infect. We do grow more highly susceptible cultivars, and then sometimes there is a chance that that this pathogen really thrives well because people are not removing fire blight strikes from the orchard as diligently as they should, because the fungus inhabits the dead tissue of the fire blight strikes, and that's where it also can overwinter. And the orchard sanitation, if it's not applied to remove the decayed fruit from the orchard floor and mummies in the canopy, it seems that really contributes to this pathogen to survive better. So if you have a grass strip below your trees, it seems that that's actually good because the grass strip allows the fruit actually to decay better, because the activity of different flora and fauna there is much higher, so the fruit degrades faster than the actual herbicide strip. They just stay there mummified. So the late maturing cultivars might also have issues with not enough residue, fungicide residue late in the season, and that seems to contribute allowing this pathogen to infect. And then we also might be having issues with people implementing or activating irrigation on time. Usually it's the time when people actually see the trees being water or drought stressed that they turn on the irrigation. And that's too late, because the drought stressed trees have compromised immunity, especially the fruit. And that seems to contribute to the infections on the fruit, because the immunity of the fruit itself in the skin is compromised, and that allows not only different physiological issues to happen, but also for fungi to be reactivated and infect. So we have many open questions about bitter rot. Do we have this dormant infection? So sometime, during the petal fall, there's definitely reports in different parts of the world that these fungi infect during the time. They can infect as early as petal fall or bloom, but they stay dormant or quiescent up until the time there's certain cues that they reactivate them. So we don't know whether that happens in New York, but it's highly likely. We do not know, and there's no research whether heat fruit injury contributes to these fungi, to infection. And sometimes, we can't correlate, or sometimes you can correlate also sunny side of the fruit that is very exposed to the sun and stressed to the incidence of these diseases. And also, we just don't know which species are present in New York, and the species can vary widely in their biology, but also sensitivity to fungicides. And different farms have different management practices, so that can also contribute to the different incidence depending on how you approach your management. So definitely, there's many different things that can be researched here, but what we wanted to do is we just wanted to see what we have in New York, and this is just a small overview of these two groups that you see here. The blue one at the top, called Colletotricum micutatum complex. This is the group of species, okay, from the same genus Colletotricum. So it's a complex of species, meaning there's multiple species within it that cause bitter rot. And the ones in yellow that I highlighted here, that are in the blue group, which are more accustomed and more prevalent in cooler regions, those yellow ones are the ones that are so far reported that cause bitter rot in different parts of the Eastern Board and states. And we have not found these ones in the white. But the Colletotricum gliosporoides group, the red one, that is the second in line, is more present in warmer regions. And the first four also have been described on palm fruit, but we didn't know up until just this past beginning of this July which species we have in New York. And that's what my research lab investigated. I'm going to show you that in a minute. So, but before I go in and explain that, I just want to give you the brief justification of why do we need to know the causal species. Well, research has shown that if you look at the Colletotricum micutatum complex of species, that first one that is more accustomed to cooler regions, we do see more tolerance to fungicides in that group versus the other group that is more present in the warmer regions of the world. For instance, the orthopsin and raleigh might be definitely not working as well against the Echinacea complex species, such that's your Colletotricum fiorinae as a species, whereas these other ones will be more effective against the other group. So we don't know too much about the tolerance to these fungicides between different species. We don't know which species we have in New York. We also don't know what the susceptibility is to the DMI fungicides, such as raleigh, one of them, and also there is whether we have resistance to the QY fungicides, the FRAC 11 group, which is your Pristine, Marivon, and Flint, for example. And then the new class of fungicide SDHIs, such as Frontelis and Marivon, and these different fungicides, we just have preliminary data from different other parts of the world that they are hit or miss. Some of them are effective, some of them are not effective in certain species. So it really depends. And then if you go in different parts of the world, you have variation between those parts of the world in which species you have. So just if we go to Pennsylvania, you have a different complex. If you go to Kentucky, you have a slightly more different complex than us. One thing that we do see is the Colletotricum micutatum complex species. They grow at the temperatures below 41 Fahrenheit, whereas that's not the case with the species from the gliosporoides complex. So they definitely, based on research, differ in their susceptibility to fungicides, and we just don't know how they differ in the other factors, such as ecology, what are their temperature requirements, in their virulence as well, and that's like an open field that we wanted to go and investigate. We sampled these different farms that had issues with bitter rot, and with 2 of them on Long Island and most of them in the lower Hudson Valley. We sampled roughly 16 farms in the Hudson Valley, and we have 5 or 6 more farms that are outside of the state in Virginia and Pennsylvania. And we collected around 400 isolates, and then when we did the isolation, we have conducted a deep sequencing analysis that led us to identify that 60% of our collection that we have collected from these farms, 60% of them were the bitter rot caused by Colletotricum fiorinae. That's the species in white, font up there. 25% of them were caused by Colletotricum chrysophyllum, the species that has not before been reported on apple. So we are a little bit dumbfounded with that because we did not expect that species. It's known only on banana so far and on cashews, okay, in the really tropical and subtropical parts of the world. And we have found it here on apples. And so that's the species from that group that I mentioned to you is more prevalent in the warmer regions of the world. And then 12.5% of the samples consistently throughout the 2 years, we have seen this new species that we named in our research recently as Colletotricum noveboracense. The noveboracense is the name of the species that is Latin translation of New York State. That's the way how we wanted to describe it. It's a pathogen within that group of more warmer prevalent species. It falls in that group, and we have found that one. That was the big surprise. We did not expect to see at least 2 new species for this host, and 1 new that is not as described before. We expected some other species like Fructicola or CMNC and other ones that are described in different other states like Kentucky and Pennsylvania, but lo and behold, that was not the case. So this just came out just several days ago. So in a journal, we just we published this description of the species, which is something that really contributes to other researchers a lot. There's a lot of people in different parts of the world working on these, not only on apple, but on mango, on all these other crops, field crops, fruit crops, name it, they have it. And also the nursery crops as well, flowers and stuff like that. So it's really a huge field of research. And so we're happy that we were able to report this, because we believe this is going to contribute to the better way of management in the future. So talking about the varieties, this is a slide I mentioned briefly the varieties that they contribute. If you grow more susceptible varieties, you might be having more issues with bitter rot. So this is what my colleague from University of Georgia has in his presentation, Philip Brannon, who actually just summarized briefly this study from Alan Biggs that showed that if we look at this, they looked at one species called Tetrachomycetatum from that first group that likes more cooler regions. You can see the Honeycrisp and Pristine in their research was very highly susceptible, or the most susceptible, including ginger gold, whereas the least susceptible would be your Fuji, or moderately would be maybe Golden Delicious and Gold Rush. So we did not have any data for New York in terms of this susceptibility, and we did not even know whether we should expect to see any differences. But this is what they found, in Virginia and other parts of the eastern board. And what we did is we took 2 species. So the one that we tested is the one called Colotaticum furinia, that is the most dominant that I mentioned to you. And then we tested Colotaticum chrysophyllum, and it seems that there's a little bit more virulence or aggressiveness of the chrysophyllum in terms of the size of the lesion that it causes on these different fruits that we inoculated. We infected them with these fungi. So it seems that there is a little bit more virulence in these species that are more prevalent in the warmer regions. In comparison to the Furinia, it seems that, overall, if you would average these across different cultivars, that they have a little bit less virulence in them. So they can vary in their, ability to cause a disease. Also, what we did, we tested this at another stage. So the first image I showed you was when the fruit was about an inch size in diameter, and this is the near maturity. So these fruit all were picked up around the first week of September, when Honeycrisp harvest time is. And we also can see the similar trend that overall that these species like Chrysophyllum, which we chose here as a representative, is slightly more aggressive in comparison to the Furinia. It has larger size of the lesions that it causes, that to us indicates that they differ in their biology and in ability to cause a disease. So we also went on and then tried to compare all 3 different species. So these 3 bar colors that you see, the white, the gray, and the black, are 3 different species. And within each cluster of these 3 colors, you see different cultivars. So ranging from Fuji, Gala, and Red Delicious, and Golden Delicious, and Honeycrisp. And you can see here that we have statistical differences in their ability to cause a lesion or the rot on fruit, okay? And, specifically showing here that maybe on Gala, the Chrysophyllum and the Novaburracensi, the species that are more accustomed to warmer regions, are more virulent on Gala than maybe Furinia. And you can see that each of these different cultivars have slightly different responses, and they are statistically significant. So we reasonably believe that they have different compatibility with different varieties because they have different pathogenicity related characteristics. And so it's important to know the species that you have in your orchard, because then the management can be done better based on this. In terms of the life cycle of this pathogen, so this is a simplified life cycle. If you start from the bottom of this graph here, or maybe on like a cartoon that I just pasted there, you'll see that the pathogen overwinters in the buds and mummified fruit and in the cankers on the wood, and also on the brush that is left over on the ground, but also under the bud scales of the alive buds. And in the spring, the rain allows the pathogen to splash its spores from these mummified fruit and cankers and buds and onto the flowers and eventually on the fruit during the summer. And then the pathogen forms new generation of asexual spores. And all these organs that are infected that you see in spring and summer can drop down on the ground and allow the pathogen to bridge over the time up until the next season. And eventually the fruit also drop, the flowers and the leaves can drop and also the brush that you leave there that is left on the ground on the orchard floor can allow this pathogen to overwinter and carry over to the next season. So in general, the survival is definitely multi-pronged. There's multiple sources of inoculum. There's also very good evidence that's going to come out soon in a research paper that this pathogen survives very well on many different forest trees as an endophyte. So meaning it just dwells there in the leaves, does not cause any symptoms, and the tree has it, and then eventually just spills over into the orchard. The spores the pathogen releases in the spring and summer, they are rain splashed, and they can be dispersed throughout the whole season. There's a huge infection pressure, meaning there's a lot of spores to be released both from the forest and also from the inoculum sources within the orchard, and that continuously is happening through the season. Sometimes as little as 5 hours of leaf wetting is enough for infection to occur. So basically those long warm wetting events increase the infection severity. And when you see it, that means that the fungicide residue was not there, but also that the weather was very conducive. The fruit usually get infected. They can get infected as early as bloom, but we don't know whether that happens in our conditions yet, and people are still investigating that on the eastern board. But majority of infections usually happen mid to late season. So basically right about now up until the end of the season. That's when people start, noticing it. And we see these severe epidemics early in the season when we actually have conditions right about like we have right now in the lower Hudson Valley, very warm weather that is followed by rains. So that weather is allowing the primary infections to allow, to happen, and then those infected fruit form new spores that then they carry out and they actually conduct secondary infections, and then we have a big headache because of that. So this is another just cycle that I just wanted to show you. This is the way how the reproductive structures of this fungus look like. The bigger circle that you see all around shows the sexual stage of the fungus. So some species are known and reported to have sexual reproduction. So on the outer left side, you see perithecium there, the fruit, the pear fruit looking like a fruiting body that forms ascospores and they can infect. And if you go to North Carolina, you'll find from Sarah Villani that she is actually monitoring these spores to detect when they are actually released to allow infection. But I don't know whether we have them in New York. But the inner circle that you see on the right side is the reproduction with asexual spores or conidia, and they can, the bilirot fungi can have multiple generations of conidia. And one thing with this arrow that says quiescent latent period, I just want to emphasize to you that this pathogen as well seems to be dormant after the infections during petal fall, and it can dwell on the skin that you cannot see the symptom that whole time up until the point when fruit starts to mature or certain other stress factors play into the fruit immunity system, dropping the fruit immunity system, and then the pathogen reactivates and infects, and you can see the symptoms. So that's the problem also with this fungus as well, that most likely it has that quiescent latent period as well as the black rot fungus. So what we wanted to go and try to do is see if we can actually isolate this fungus in the middle of January and see if we can, how many buds can actually contain this fungus. So this is a summary from 2 years of data. And these are the varieties that we tested, that you can see on the left side in the first column. If we would summarize all that, we found that out of 100% of the buds that we collected over 2 years, we found this pathogen, these pathogens overwinter at 15% of the buds. So if you had infection last year, there's a high percentage of chance that inoculum is going to be surviving really well under the bud scales of your regular fruit buds that are going to open next year. And out of those 15%, we had 10% of them where the species from the Gliosporides complex were. Like specifically, I'm telling you about those 2 new species that we detected, so the ones that prefer warmer regions. And then the 4%, so the smaller percentage of them, were the ones containing species that is more prevalent, the Fiorinae species that is more prevalent to the cooler regions of the world. So that's kind of like interesting, and we did not expect that. So definitely it seems that in New York, the buds can be a source of inoculum, but we need to repeat this a little bit more, and with more sampling. We just had 2 locations here, so we want to do more with this. In terms of the fungicides you can use, these are the main groups, or the FRAC groups that I would mention to you here. The FRAC is an acronym that stands for Fungicide Resistance Action Committee. There's a group of scientists that work 24/7 to prevent that the resistance to fungicides occurs. And basically, they actually recommend these codes that you see here, such as code 11 for QI fungicides. The MBC thiophanate methyl belongs to the thiophanates, which is your MBC group 1, DMIs, FRAC3, and SDHIs. And I mentioned here omega-fluazinan, which is a new group of the fungicides, and we don't know whether it's effective against bitter rot. But if you look at these first 5, those are actually the fungicides that you are dependent on to control bitter rot, especially on the QIs. So we test these fungicides in the lab. So this graph shows you 3 different species: the Fiorinae that loves more cooler regions, and the black and the gray one that love more warmer regions. You can see there is a huge variability how they respond to different fungicides. So for instance, difenoconazole, which is the far left one, seems to be quite effective, but there is still difference in different species how they react to different fungicides. And if you go to benzoin diflupyr, which is your 4th from the left, you can see here that in this case, there is more tolerance of the Fiorinae species to that fungicide than maybe the Novaburracensia or Chrysophyllum have. So definitely, this is an important factor in the way if you want to manage bitter rot in general. Which fungicides you combine in tank mix and how do you alternate them seems to be very important. So in the orchard, in the way how I would advise people to approach bitter rot control is that you should always have an assumption in your mind that apple fruit are susceptible at all growth stages of growth, but definitely more when they start to mature. That if you have weather like we have right now this July, when we have very warm and hot days that really precede warm weather events, that those are going to be the prime infection events that are going to allow infection by these fungi. Never delay your rot sprays beyond the 10th July. But keep in mind that infections can start as early as 1 to 2 weeks after petal fall, and they can the infection period, meaning the spores can be produced by the fungi all the way up until the harvest. So it's a completely contiguous block of time when you can have infection. If you do spray, try to align your QOI sprays, so your Pristine, your Flint, your Sovran, or your Maravon. You should try to align them before the warm weather events, because those are the most severe ones that you should protect against. And if you can, you should keep up with the rain. If the rain is frequent, like it is in these past several weeks, you should definitely spray on an interval 10 to 14 days. When you get to more drier parts of the summer, you can stretch to 21 days. But if you get 2.2 inches of rain, it doesn't matter when you sprayed. You have to go out and repeat that spray that you did, even if it was 1 or 2 days before, because you lose that residue on the fruit that really protects the fruit. So that's what you need to maintain on the fruit. Some late maturing cultivars, like that mature up to the beginning of November, they have to have continued residue protection with fungicides up until the harvest. In general, what I wanted to say is that even though that we are dependent on these QI fungicides like Flint, Pristine, and Maravon, and Sovran, and Luna Sensation, you can definitely always tank mix with Xyrem and Furban and Captan, and you should, for anti-resistance management strategy that basically prevents potential development of resistance to QOIs in these fungi, which we don't want to have. And Topsin, you should bear in mind, and the DMIs do not seem to be very effective against the bitter rot fungi, which is kind of something that really bothers me right now, because if you look at the data from Georgia, which I'm going to show you right now, it seems that it's very effective. Inspire is very effective against bitter rot. So we don't have the data. We're going to try to get the data. So if you use Captain alone, besides mixing it with different fungicides, you should go with higher rates, 3 to 4 pounds, or between 2.5 to up to 5 pounds of Captain of the 80 WDG formulation, because you can't rely only on a low rate when you have high infection pressure. You can, even with a high rate, you can lose the battle with bitter rot, under the high infection pressure. So you have to increase that rate if you're using it alone. So basically, the overall idea would be to combine any of the QI fungicides, the FRAC 11 group, with any of these contact fungicides such as Captain or Xyrem or Furban. And they're going to be, Captain and Xyrem is going to be effective against Sooty, Blotch, and fly speck as well. They're going to help you against it, but primarily the biggest muscles are going to be by the QI fungicides. They are also effective against Sooty, Blotch, and fly speck. To prevent any storage incidents of bitter rot, what we would definitely recommend is that you keep up that residue up until the harvest, and that if you can, you cool the fruit after you pick them immediately at 40 Fahrenheit. That seems to slow down the development of bitter rot. So if you can do that, if you're able with the capacity of cooling, you should do that because it helps a lot. And then in post-harvest mode, you can apply scalar fungicide for dioxanil, but it can only be effective against fresh infections, meaning the infections that just set in before you harvested, not the ones that progressed already. So there is some benefit of scalar there. It seems to be effective. So one thing that is difficult, if we look at this strategy for bitter rot control is, and many people complain to me about this, that we have to limit the number of QI applications to 4 sprays per growing season. And the reason why I am really, really pushing for you guys not to use more than 4 sprays is because we do not wish, for these Coletotricum species that cause bitter rot, we do not wish them to become resistant to QI fungicides. And knowing the history, and some farms applying 6 to 8 sprays of QI fungicides throughout the summer, that's just calling for trouble in the future. And if you can, you should probably back off from using any of the QI fungicides, Luma Sensation, Flint, Pristine. Not use those early in the season for powdery mildew. Just focus primarily on the DMIs, primarily on Rally, Rhyme, Indar, and Procure, and finish your early season that way, and then save these 4 QOIs for later in the season for bitter rot. That's the goal that we have to achieve, if you can. So, for instance, if you had issues with powdery mildew last year and you don't want to have this issue, you should maybe start a stage earlier at tight cluster. Never miss your pink and petal fall spray with DMIs, and then you should be okay with powdery mildew without using the QOIs. And then if you do deliver during the summer, your Flint, Flint Extra, Sovran, the QI fungicides, Pristine, and Luma Sensation, you should definitely do it just before the hot summer days followed by rain. I know that that's not optimal every time because there's probably more infection, more like rain events that can trigger the infection. But these 4 applications of QIs per season, you can alternate them with these middle group of Captain with Inspire Super, or Captain plus Prophyte, or Captain plus Topsin, or Furbam and Xyrem. So these contact fungicides such as Captain, Xyrem, and Furbam should definitely be tank mixed with the QI fungicides when you apply QOIs, but then you can also use them standalone at the high rate, and also you can mix these so that you can bridge over that time up until you come to the harvest. So usually we end up having between 6, 7, 8, or 9 cover sprays, depending on the year, depending on the amount of rain that we get and the amount of rain events. But in general, I would advise you to use mancozeb early in the season. That is going to aid you controlling the scab and rust. Then switch to the Captain schedule for the cover sprays. If you do opt for using, Prophyte, you should definitely be encouraged to use Prophyte because this product does not cause any, resistance. It's not prone to causing resistance in these fungi. It actually acts by boosting the immunity system of the fruit and the plant. So it does bear a benefit there, and it's also effective against, it can help against Sooty, Blotch, and fly speck. When you alternate the fungicides, you should definitely keep alternation, because in this manner, you should apply, maybe you could apply 2 QIs in a row, then you should not, apply the third one in a row. You should definitely do a different combination. And some of these combinations that I mentioned here, you can apply 2 in a row, and to bridge over up until the harvest. When you get the 2-inch rain event, you should definitely respray. So whatever you tank mix, bear in mind that you're broadening the spectrum of efficacy against these fungi, and what broadens the efficacy also helps you prevent the resistance to move into these fungi against the QI fungicides, which is exactly what we want to achieve. So we are actively working to expand this in the list of options for you in the summer. So we have a big project funded by the Farm Viability Institute this year, and the next year is going to help us maybe see which fungicides might be the new ones that we can implement in the future. And just to show you that the situation is not as dim in the New York as it is in the southern part of the Eastern Board, this is just a list of the treatments that Philip Brannon from University of Georgia has compiled of what is actually legally allowed to use. And I'll focus your attention to the bottom of this slide, where the cover sprays start. So the second cover starts down there with yellow, and then you switch to the white cell there with cover spray 3, 4, 5, 6, and 7. And one thing that really is interesting is, yes, you can see there that they apply mancozeb with Prophyte, and they also mix it with Top Guard. They have Maravon and Captain and Prophyte alone. And they sometimes do 2 or 3 in a row of these combinations, okay? But what really bothers me is, look at how many cover sprays they have in total. In the southern parts of the United States, they go up to 21 covers during the summer. So talk about intensive production of apples there. And that's the reason why this is the case with them, is because they have a longer season there than we do. So they have these many options that are shocking to me, such as Inspire Super. They cover 9, if you can see here, they recommend Inspire Super. It seems to be effective against the glomerullar leaf spot, which is the leaf form of this disease of the bitter rot. And then they alternate in also the Omega, the fungicide that is new that I just mentioned to you, which is from FRAC 29 group. And they also alternate Inspire Super. So based on our experience, Inspire Super, it seems not to be effective against bitter rot, but they have some data to prove that it is effective in the southern parts of the states. That might be because they have different complex of species in those states versus us. But we usually end up season, maybe in the worst case scenario, with 9 covers. They can end up season with 21 covers. So that's quite intensive. So what about organic orchards? And I know these questions come up a lot of times. Yeah, that is a problem. Okay, bitter rot is the biggest problem after rust that people are worrying about. Dormant copper is going to help you, but it's just going to reduce the overwinter inoculum. It's not going to go and help you with the seasonal infection pressure. If you do apply sulfur, maybe it can help, but you should use the fine ground sulfur, and you have a limitation that you can apply it up to petal fall, because after petal fall, sulfur, too much sulfur actually can reduce the photosynthesis, and you don't want to have that. You want those fruit to grow from the photosynthesis of the leaves, and it can burn leaves if you apply it to temperatures above 90 Fahrenheit. Cueva and Bacjex2 can be applied at low rates, but you might be having issues with fruit russet. Some growers tell me that's not the issue for them, because if it's organic, they can sell it. And then the dependence on liquid lime sulfur, you could mix liquid and maybe control a little bit bitter rot with liquid lime sulfur in combination with low copper rates. However, the liquid lime sulfur has a big issue with it. Usually people use it for Sooty, Blotch, and fly speck control, but you have to use it so frequently that in some cases, the oil that you add to liquid lime sulfur can bring this mix to cause injury on the fruit. And that might completely increase the amount of rots that you can have there. So that's the issue with organic production. The list of materials you can use is quite narrow. If you do have an organic orchard, the cultural practices are your best friend. The position where you plant your orchard has to be such that you have perfect aeration or air circulation through the orchard. Definitely have great sanitation to remove anything that might carry the inoculum there. When you do tree pruning, make sure that you do it perfectly so that you have good light penetration and to allow fast drying of the leaves after the rain. And you should avoid planting on poor sites that are low. And if you do have irrigation, that's definitely something that you have to have in these orchards so that you can prevent the drought stress in the trees, which kind of seems to exacerbate bitter rot. There are differences in the cultivar susceptibility, and I just listed you here some varieties, okay? But depending on which pathologist you ask, they're going to go and say that there is no resistance in these commercial varieties that we depend on to sell the fresh fruit. There is some germplasm out there that has been discovered that bears the genes for resistance, but it has not been bred into the commercial cultivars yet. If you do harvest the fruit, always try to cool them as much as, as quickly as possible below 42 Fahrenheit, because that's going to slow down the growth of these fungi, specifically of that group from the warmer region that I mentioned to you. Some tests have been done with Cueva and double nickel, and I'm going to show you these graphs. So this is the Keith Yoder's work that has been done at Virginia Tech that shows that if you apply overall the OSO, the new fungicide, which is polyoxin D active ingredient, it seems that to control this, to control overall the rots, and if you look at the right graph, it seems to control with 9 cover sprays from petal fall till the 9 cover. If you apply this, product, it might control bitter rot pretty well. However, I'm not sure which species they have in Virginia, and also whether this also fungicide would be effective against the species we have in New York. The reason why I'm putting this slide here, okay, which shows the white rot on the left graph and the alternaria rot on the right graph, is to show you just that something that works against bitter rot might not be effective against white rot. So it's highly specific. Like in this specific case, the new fungicide that you see as a green bar on the left might not be effective against white rot at all. Whereas for alternaria rot and bitter rot, it might be very effective if you apply it as frequently enough. So these new chemistries that are coming out might be completely hit or miss depending on which rot you might be having issues with. And finally, this is work from Jeff Alejandro in Western New York, which shows that if you have a spray program, which is the blue bar that you see there flattened as a pancake, that's your Cueva and double nickel mixed in with alternating spray program with also this new fungicide called polyoxin D. And it seems to be working quite well in this year. So it was very comparable to the grower standard. So hopefully in the future, these get labeled or maybe used more by the organic growers. So I'm going to switch now and talk about the Sooty blotch and fly speck. This is an issue that keeps on happening in the years when we have so much rain, like 2017 and 18. And it seems that we had it quite a lot in the valley. One thing that I would just definitely urge you to do is use the models in NUA for Sooty blotch and fly speck, which allows you to put in your petal fold date on average for your farm, and then it will give you the estimation how many accumulated leaf wetting hours, how quickly they are accumulating from petal fold on. And once you reach this red zone that is visualized here in this model, so this model is free, keep that in mind. Once you reach about 190 hours, which is here on this graph, on this table, it's on July 10th. Just before that, you should apply a material that is very effective against Sooty blotch and fly speck. And as I mentioned to you, QOIs really are the most effective, and also Inspire Super is very effective. So you should probably apply something just before that, 190 hours as a cutoff point when we expect to see infections from Sooty blotch and fly speck. So this is a free model you can use, and it helps a lot so that you know approximately after which time you should not delay applying these fungicides. And as I mentioned to you, 10th July seems to be the time that you should also not miss for rots. So in this specific here, this is at least for our location here where the station is. There's other models for prediction of Sooty blotch. In this specific case, if you are a subscriber for Rimpro, and if you are working with me on using the Rimpro model for scab prediction, that cluster of models, for the same price you paid for the apple scab model, has a model also for Sooty blotch. And one interesting beauty of this model is that it can give you this top graph that you see there that shows you in this a green patch up there when the infection has actually occurred and there is actually pathogen developing, but you can't see the symptoms. You can't see that it's actually there on the fruit. And at the bottom, these red curved lines that you see up there, those are the infection events that happened. So the pathogen starts developing, and in this specific case, you see that infection actually happened on June 28th. So it seems that this year is going to be a wonderful year for Sooty, Blotch, and fly speck. At least this was in 2018, and I'm going to show you right now how this kind of a graph looks like for the 2020. But after this big green patch you see up there, there's two gray ones on the right side in the top graph. Those are actually the ones that you can see. Those are the spots you can see. So then the model can give away when this fungus is present there and growing, unless you applied the fungicide and you suppressed it. So this is a cool part of this model that newer model does not have. And this is, I was going to show you here this, again, the first infection in 2020 happened on June 28th, and we had another infection on July 4th or July 5th, depending on location. So just making sure I tell you that, and that these models are available. This one is paid, but if you do pay it for scab, it does pay off also for Sooty, Blotch, and fly speck. We do test fungicides for Sooty, Blotch, and fly speck control. We tested one for a Canadian company. It is called AF Global, organically acceptable product. It didn't seem to control Sooty, Blotch, and fly speck, but I wanted to divert your attention to the treatment number 5 on the right side, which is the grower standard. This is 2 applications of Captain and mixed with Prophyte. Then we had 4 applications of Captain and Topsin. Then 2 applications of Captain alone. Oh, actually Captain with Maravon, and then 1 application of Captain alone. So this totally gives you 6, 8, 9 covers that we have done. And the reason why I'm showing you that is because if you look at these graphs, okay, this is how the Sooty, Blotch, and fly speck incidence looked like. So the yellow bar or the light brown bar is your Sooty, Blotch, and the purple is your fly speck. So we have looked at the incidence of on Macintosh fruit, at harvest, and the right graph is 2 weeks after harvest. And you can see that the number 5 treatment, which is on the total right of both graphs, shows you that it's definitely working the best. So, and we have repeated these results not only on Macintosh. We have graphs that show you the the treatment number 5, was best on Ginger Gold as well, and also it was very good on the Golden Delicious as well. So these fungicides are very effective, and these new fungicides that I tested, the number 2 and number 2, did not really work well. So sometimes this happens, and but that's why the research is there, to see if we can actually have any efficacy or not with these fungicides. So, and then finally, I'm going to close with the last thing, the last pathogen that I believe is something that is of a problem and emerging across the eastern board, anywhere from North Carolina all the way up until the south of Canada. People have issues with defoliation from this fungus. It's caused by a fungus called Marssonina coronaria. These are the spores that you see in the central image, the conidia, the asexual spores of the pathogen. And on the right side, you can see roughly 40 to 60% defoliation, roughly around like mid-September. And that causes a lot of distress for people who have pick your own operations. They don't want to have that. And it's actually caused by this fungus. And this was in 2017. We had issues with it in 2018 as well. And one thing that was difficult for us is to work with this fungus. It's just we have to get it out of the leaves and have a pure culture so we can work in the lab with it. And in Switzerland, this is a big issue on apple, that also it causes blotches on fruit. So in the northern part of Italy and Switzerland, it can cause symptoms on fruit. And we thought, okay, let's go and try to find symptoms on fruit. And we did. That's the way how we were able to get this fungus out and work on it. And we teamed up with Carrie Peter from Penn State, who's my counterpart in Pennsylvania. And we were able to confirm that we do actually have this pathogen, Marssonina coronaria, in New York. We don't know whether this pathogen has always been here or it's a newly emerging pathogen, but we didn't have it in the past as an issue, but now it seems to be present in the last 3 or 4 years. It seems to be in the orchards, and it can cause blotches on the fruit as well, not only on the leaves. So when you have defoliation in the leaves, you might be having spots on the fruit. And sometimes when you get these fruit picked up and stored, when you get them out of the storage, they can have these spots. So call me if you do, because we would love to get our hands on these fruit so we can actually get the fungus out and work on it. We also collaborate with Switzerland pathologists. Thomas is the one that we work in the Organic Institute of Switzerland. He's our good collaborator there. We're working with him on trying to determine how did this pathogen, become to be such a problem in our region and in their region as well. There are issues. People call me and get confused. What are different symptoms? So if you can focus your attention to the middle of this cartoon, you can see how the symptoms from Marssonina leaf blotch actually look like. But on the left is not Marssonina leaf blotch. On the left, you see necrotic leaf blotch, which is a physiological disorder typical for Bold and Delicious and some other varieties. The blotches are bigger and irregular, whereas the Marssonina leaf blotch spots are small. And then on the right side, you see the frog eye leaf spot. So there are certain differences that you should look at, and it's not everything is Marssonina leaf blotch. And then eventually, when the leaves start dropping off, you see these frondy edges of the Marssonina leaf blotch. So it varies also depending on the time when you look at the symptoms. The reason why this pathogen is really becoming an issue is because we have had times in New York in 2017 when the rain was so frequent that over like 40 days we had wet conditions. And that seems to be based on this research by the Korean group in 2011. Just shows you, depending on how many moist days you have, how much infection you have. So the 3-day moisture on the leaves can lead to 72% severity on the leaves if you didn't apply any fungicide. So the more moisture you have, the more disease you have, and that explains why in June and July, the rains that happened in 2017 led to this pathogen to really set in really hard in different apple cultivars. So not only us had the infections, there's Pennsylvania, Wisconsin, and Virginia, and North Carolina reported it, Canada as well. In the past, this pathogen has been described in China and Korea and in European Union and Brazil. Seems to be unusually wet summers that actually cause this pathogen to occur. Especially organic orchards are heavily damaged by defoliation, and there's no labeled fungicides, at least in New York, that you could use against Marssonina. This fungus has low susceptibility to copper, so the copper might be the issue, not being effective against this in organic orchards. Varieties that are susceptible are listed in red there. I'm not going to go and read them to you, but I want to mention to you that this is just preliminary based on what we received as samples throughout the last 3 years. Those are the samples that we have seen defoliate. Those are the trees or varieties that we have seen defoliated by this fungus. We do have varieties that are moderately resistant, okay, such as these Chinese varieties that you see in blue there, and also Pinova and Akane, but they're not as prevalent or at all existing in the northern United States. Just briefly to mention to you that this pathogen is really important in Asia. They even have a breeding program to breed the resistance into rootstocks, not only varieties, but also rootstocks against this fungus, because it causes huge issues in rootstock liner production. So it can defoliate liners, and then you lose the yield of them so that you can actually use them to graft on. If you can, if your orchard is too small or is smaller, on the smaller side, we would definitely recommend you to apply urea at 40 pounds if you're a conventional grower, and dissolve that in 100 gallons so that you can actually spray that on the leaf litter or just before leaf drop on the trees. Or if you are an organic orchard, a grower, then you should use dolomite lime at 2.5 tons per acre, because either of these options allow degradation of leaves much faster, and it seems the pathogen overwinters really well in the leaves. So if you can prune well to get good air circulation to dry the leaves faster, that's good. And then there's different fungicides that different research shows that they are effective, like mancozeb, the Mataram, Topsin, and diphenetmethylamivir seem to be effective. So, and also the DMI fungicides, such as Spy Super, might be effective. But we don't have any efficacy trials for this fungus yet in the valley. And some research is being done in Pennsylvania, so might bring up to some options in the future. So it seems not to be a widespread problem, but more and more people have issues in the years that are with unprecedented amount of rain during the summer. This is the life cycle of the pathogen. If you look at the left side middle image, this is the leaves, how they look like, and the black and white image shows you that pathogen overwinters on dropped leaves. And that's where we want to apply the urea or dolomite to get dolomitic lime to get the degradation of those leaves. The pathogen can form an asexual form, a cervuli, a disc-like shaped fruiting bodies that form sexual spores. And also the asexual spores called conidia in the cervuli survive. I'm sorry, I made a mistake. The sexual stage has apothecia. The pathogen then survives on the leaves both as apothecia or in sexual stage or conidia, and then it can infect with rain in the spring. And then it has multiple cycles of conidia that keep on infecting if you don't have fungicide coverage, all the way up until the new leaves drop and you get defoliation of the trees, and that's where the pathogen survives. We don't know whether we have a sexual stage, the apothecia in New York, and many locations in the world think that pathogen survives only as asexual stage on the leaves. If you pay Rimpro for subscription for scab, again, another model for the same cost of the scab model, it is really great. You can also get prediction for when the first infection during the season can happen from Marssonina leaf blotch. So it's very useful. It works in the same capacity as Sooty, Blotch, and Speck mode, gives you the indication when the first infection happened, and you have to apply something that's very effective against Marssonina just before that infection, and you should be able to control it pretty well. We work with this fungus, this pathogen in the lab. We test different fungicides to see how effective they are. We do see a little bit more effect of difenoconazole versus other fungicides, but this is just preliminary. Still, we're still working on it. And this is the lab scenario where we mix a medium on which this pathogen grows in the lab. And it's not the same as testing them in the field. So that's the plan in the future. And we're going to see if we can do that. But, you know, we want to be ahead of the curve, prevent any potential resistance in this fungus to fungicides. And that's the reason why we're screening these fungicides on this fungus. There's research in Switzerland, actually in Germany, that has been published recently for organic producers. This is an efficacy trial in 2016, and at the bottom graph in 2017. How did 10 to 12 spray applications of each of these products that you see at the bottom of the graph, how did they actually turn out if they sprayed from roughly about like 10th to 12th June up until 30th August. And the most effective you can see that are consistent both trials is this mycosine, or the acidified clay, and mycosine plus sulfur. And in this case of 2016, they had good results with copper funguran progress. So it seems that there is some options for organic growers you could use, but the mycosine that is listed here is not allowed or registered in the United States because it has a high content of aluminum there, which might, you know, bring up to contamination of the soil. But it seems that organic growers are certified in Europe to use these products that might be very effective on this very intensive schedule of spraying to protect against Marssonina leaf blotch there. So this is, I'm just mentioning to you the way how the clays work. Mycosine works in the way that the clay actually has aluminum content that somehow, you know, basically is inhibiting spore germination on the leaves. And by increasing the aluminum content, that spores can't germinate. The clay also allows to absorb water. You know, the clay absorbs water, and that's the way how it actually reduces the wetness after the rain, and somewhat, you know, reduces the risk of infections. So that's one way how the clays can help. But not all the clays are the same. This specific one may be very effective, whereas maybe kaolin or some other clay might be completely different properties. So it's not the same if you use maybe something else that is registered. So with this, I'm going to close. This is my last slide. I have collaborators which we work with. I mentioned to you we're testing fungicides for bitter rot in the next 2 years, including this year. We're hoping to learn more about Clotonicus species by sequencing the genome, by looking at the DNA sequences and, you know, what's the makeup of that sequence and how can we actually use that information to better manage the bitter rot as a disease. We don't want to have resistance to strobilurins, so one way we can use the genome sequence is actually to search for any insertions of transposons that maybe allow mutations that can bring to the resistance to the strobilurin fungicides, the FRAC11 group that is the most effective group against these fungi. We have a specialty crop block grant also that is going to allow us to develop PCR methods, so the molecular-based detection methods for these species. So if a grower comes to us, we can have a result within 24 to 48 hours, 24 hours, we can have a result which species he has in his orchard. And we're working on Marssonina coronaria, trying to determine and characterize this pathogen and explain maybe potentially why this disease or the pathogen is showing up in Europe now, and it did not in the past. So with this, I'd like to acknowledge the different funding sources and many people really that I work with and collaborate. Without them, nothing would be possible, and I would be definitely willing to answer any questions if you have them. And I just wanted to show you here that that the diseases I work on really make the covers of certain journals. The bitter rot is an important issue anywhere from Ohio all the way up and all the way down to Florida and up to Canada. So feel free to ask me any questions.
All right, great. Thank you so much, Srdan. We do have a couple questions that have come into the chat box, and we also received one earlier yesterday. So we have a question: when summer rains are frequent, would you expect longer residual control of Marssonina if Rally or other products are mixed with Captan?
Yeah, that's a great question. I don't know. We don't know much about this fungus to be able to claim whether it will be a longer residual. But you know, when you mix those two, we know that a Rally might have some kickback activity, okay? It might have up to 48 hours. In the past, we used to say that DMIs might have up to 72 or even 96 hours kickback activity. But, you know, and that's kind of like in the reverse, thinking about the residual. I don't think that residual here is an issue. I think that what might be an issue is whether you applied your kickback activity material to reach back enough after the infection. And that's where the model that I just mentioned to you for Marssonina can help. If you look at the model, you can know when the infection happened, and then you can design when to apply your Rally to cover in past what happened as an infection. So you have a time after the rain event when the infection happened, up to 96 hours maybe to cover with Rally to control that infection, okay? But in terms of the residue, it's still, the jury is out there, okay? How much residue with certain amount of rain you can have, you know, on the leaves. And it's just, it's not based on research yet. The rain fastness of our fungicides is just not a field that has been heavily researched. The RimPro model that I just mentioned to you, that also has the Marssonina model in it, has this very interesting function called spray sheet, where you can actually put in your fungicides that you applied, and it would give you a simulated model on how that residue actually declines over time with rain and tissue expansion, okay? So it's just a model. It has not been really heavily validated, but it's the best thing that, you know, that potentially we have out there, even though it's not perfect, to maybe give you an estimation how long can residue last in relation to the actual material, the leaf expansion, the tissue expansion, the fruit expansion, and the amount of rain that you get, okay? So those are the three factors that really go into, you know, any discussion about residual. So we really don't know. We know that Captan redistributes a little bit better with rain, but, you know, mancozeb doesn't. So if you get 2 inches of rain or more, you know, all bets are off. You have to come in as soon as you can after that and recover. So if you really want to go and talk about residues and try to, you know, do some, I guess, testing of that model in RimPro, you're more than welcome. There's growers who are using it, and they actually say that it's very useful to them. But we don't know whether, you know, that's really reliable yet as a data, you know, source to use.
We have another question. I think you addressed this somewhat in your bitter rot talk, Srdan, but just wondering about combining liquid lime sulfur and Queva to get better organic control of bitter rot. Can you speak a little bit more on that?
Liquid lime sulfur and Queva. I think that if you do those, if you do that as a combination, you know, one thing that I do know is that Queva and double nickel seem to be a good combination that reduces the potential for rusting. Okay, I don't know much about the Queva mixing with liquid lime sulfur. You can definitely use those two, but you're going to have limited efficacy. It's not going to go and be your, your QY fungicide. You just can't achieve that level of efficacy. And the other thing that you can achieve is this perfect coverage throughout the time to be able to have the residue and fruit to protect, really well. So it's not going to be as effective, okay? And its contact, whenever you get rain, it's going to, the residues are going to decline, and you're not going to be able to protect the fruit. So the more frequently you spray those, you might be having issues, that I just mentioned to you before, where you might have some either rusting from Cueva, which is highly unlikely because that product is the least russet causing of all the products. But with liquid lime sulfur, if you do apply it, it can contribute to more chance for the damage of the fruit, especially if you add a little bit of oil there into it. So it's going to have a very limited efficacy, but you can definitely try.
On a similar note, Srdan, using just a sulfur fungicide alone for bitter rot for organic control.
Yeah, it's you can definitely try to do it as best as you can, but it's going to be the similar, as I mentioned in the former response. It's not going to be as effective. We usually also see with frequent application of sulfur reduction in photosynthesis. And what happens is when you apply too many sulfur sprays, maybe there's not too much issues with phytotoxicity from the application at higher temperature. There's more issues when you apply sulfur and you get thinning of the fruit because you reduce the photosynthesis on the leaves. And right about the time when you want to protect against these fungi, the frequency of application might not be really so friendly to the plant. So it's a double-edged sword. So and it's not as efficacious as as the other products are.
Okay, great. Thanks, Srdan. Shifting gears back to Sooty, Blotch, and fly speck. As far as the timing of liquid lime sulfur for an organic orchard, also following the NUA model, or is there a slightly different timing?
Yeah, I would use the NUA model. Okay. Maybe I would opt on a side of doing it slightly earlier. So then your usual 190 hours, because with the organic orchards, the modern infections can really backfire at you. So it's definitely something that I would recommend. Maybe right about the last week of June. I would roughly, I would calendarily say that you should start it. That would be the time when I would start it. Or maybe back off 2 weeks from what the model tells you is going to be accumulation of 190 hours, when you actually enter that red field. So yeah, the model would be crucial. And also the RimPro model for Sooty Blotch might be helpful because it can give you that indication whether the infection actually happened and which actual day the infection happened. Because remember, the NUA model is the model that gives you the estimated risk based on the accumulation of the leaf wetting hours. Okay, and we look at that, the cutoff point is 190 hours. When that happens, we know that the infection is going to happen, at least during the summer. Okay, whereas the model for RimPro actually follows the pathogen itself, and it shows that the infection happened on a certain day. So for the organic growers, it might be much more useful to have that knowledge when the infection actually occurred than when the actual accumulation of the leaf wetness hours happens. So I would recommend the RimPro model in that case.
Going back to bitter rot, Srdan, can you speak to how the 2019 season was for bitter rot problems, or if not, a lack thereof?
Sure.
And then also,
go ahead.
And then just a follow-up of the susceptibility to bitter rot of Fortune or New York 429.
Oh yeah. Well, I'll start from the Fortune. I mean, that variety is notoriously known to be susceptible. That is actually the most susceptible variety I have ever seen. Okay, they call it misfortune because of that high susceptibility to all the rots. Okay, so it's likely because it has very, very thin skin. That's one of the contributing factors, and it's so prone to bruising. So yeah, it's the most extremely susceptible variety that I have ever seen. And you don't get to see it much. So there's some farms that have it, but Fortune and then Empire are the most susceptible. So the 2019, if I remember correctly, we usually get the first symptoms of bitter rot found roughly around like this time, beginning of July. We did not have many growers contact us last year for bitter rot. I would actually love to, ask, and if somebody can comment from who's attending the seminar, if they can give me a breakdown of how did they feel the 2019 looked like. But we maybe had one or two occasions when bitter rot was a problem. Growers really learned that, the hard way in 2016, 17, and 18 that this is a big issue, and they tighten up their cover spray so they don't have it. So I don't see it much, but if I do see it, it's mostly going to be organic orchards, and I just did not myself see it much in 2019.
Okay, great. Thank you. If anybody else has any other questions, please feel free to type them in. Looks like we are getting near the end of the list. I do see one other person asked if we would have the slides available. I will say that we are planning to post a video of this. We've been recording it, and we'll have this posted within the next day or two. And any slides that Surgeon's able to give to me, we're more than happy to post up on our website.
Yeah, feel free if anybody wants to ask a question right now, and you can just speak up and ask the question. You don't have to type it.
Yep, definitely. Now that we're near the end, if anybody wants to ask a question live, feel free to take yourself off mute and ask live.
Okay.
All right. Well, I do see we are approaching 5:30. If you do have any additional questions, by all means, Surgeon has his contact information up on the screen. You can give him an email or call. Certainly myself or Dan Donahue are available for site visits if you have any questions for us. I also just want to say that, if you enjoyed today's presentation, we're having another one on precision spraying on July 20th in the evening. So, mark your calendar for that one. And thanks, Surgeon, for joining us today, putting these slides together, and thank you all for listening in.
Have a good day.